Dear students, today let us learn about plant growth and development. Let us start with the introduction. Growth is defined as an irreversible, permanent increase in the size, volume or mass of a cell or organ or whole organism, and growth in plants is controlled by many physiological processes. Plant growth generally starts with germination of seeds, which develops into a seedling and finally grows to form an adult plant. Growth in plants is usually indefinite and diffuse compared to the fixed and uniform growth of animals. Let us see a brief description of the types of growth in plants. Growth in plants may be primary growth or the growth due to mitotic division in meristematic cells of root and shoot apex, resulting in increased length of the plant body, or secondary growth caused by division of secondary meristem cells, leading to increase in diameter of the plant body. Plant growth may also be unlimited or limited. Unlimited or indeterminate growth occurs throughout the lifespan of the plant, as in root and the shoot systems of plants, which grow continuously from germination stage till the death of the plant, whereas limited or determinate type of growth occurs only for a specific period. Example, in the growth of leaves, fruits and flowers that stops growing after attaining a certain size or limit. And depending upon the type of tissue involved, growth in plants may be divided into vegetative growth phase and reproductive growth phase. Vegetative growth phase includes the earlier stages of plant growth leading to the production of leaves, stem and branches without flowers, while reproductive growth phases includes the phase in plant growth in which plants produce flowers. Next, let us see the growth curve in plants. Growth in plants is represented by sigmoid or S-shaped curve when growth is plotted against time, and it shows 4 phases. The first is the lag phase or the initial phase of slow growth. The second is the log phase, also called the exponential phase, and it is the phase of rapid growth where maximum growth occurs in a short period. The third is the diminishing phase where growth slows down, and the fourth is the stationary or steady phase where growth finally stops. Next, let us see the phases of cell growth. There are 3 phases of cell growth, and they are cell division, cell enlargement and cell differentiation. The first 2 phases, that is cell division and cell enlargement, brings about increase in the size of the plant cell, while the cell differentiation phase brings maturity to the cells. Differentiation is a process during which young actively dividing meristematic cells undergoes structural changes in the cell wall and protoplasm to form specialized cells that lose the potential to divide. Sometimes A differentiated mature plant cell may regain the power to divide and revert back to meristematic state during secondary growth, wound healing, etc. And this process is called dedifferentiation. A dedifferentiated cell may again divide and produce new cells which further loses the power of division and undergoes change to form part of permanent tissues, for example in tumor cells, and the process is called redifferentiation. Next, let us see development in plants. Development, on the other hand, is defined as the sum total of growth and differentiation. Development in plants is governed by both environmental and internal factors. The main internal factor that regulates growth and development in plants is the plant hormones, and they have both promoting and inhibiting effect on growth. In addition to the internal factors, plant growth is also influenced by many environmental factors including light, temperature, water or humidity, nutrition, etc. And among them, light is an essential one. Next, let us see the effect of light in plant growth and development. In addition to its vital role in photosynthesis, light also affects plant growth and development. The three principal characteristics of light, that is quantity, quality and duration, affect plant growth and development in many ways. The principal effect of light in plant growth and development is that it acts as a signal to initiate and regulate two important processes in plants, that is photoperiodism and photomorphogenesis. Photoperiodism is the effect of duration of light on the growth of plants, and it plays an important role in flowering of plants. In addition to flowering, the duration of light also affects many other processes in plants like bud dormancy, bulb formation, tuber formation, etc. Depending upon the requirement of day length for flowering by the plants, plants are broadly grouped into three groups. The first group is the short day plants. Short day plants are plants that require a light period shorter than a required day length to flower. The second are the long day plants. Long day plants are plants that requires a light period longer than a fixed day length to produce flowers. The third one are the day neutral plants or DNP. Day neutral plants are plants in which flowering is not affected by the length of light period, that is, they can produce flower in any photoperiods. Next, let us see photomorphogenesis. Photomorphogenesis may be defined as light-mediated development where plant growth patterns including differentiation, morphogenesis and organogenesis respond to the light spectrum, that is the red, blue and ultraviolet wavelengths of light. Photomorphogenesis occurs mainly in three stages of plant development, that is the seed germination, seedling development and the switch from the vegetative to the flowering stage or during photoperiodism. Next, let us see light perception in plants for photomorphogenesis. Plants have several types of photoreceptors for perception of different parts of the light spectrum, ranging from near UV wavelengths, that is from 280 to 315 nanometer to far red region with a wavelength of 750 nanometer. The photoreceptors contain a bound cofactor known as the chromophore, which absorbs light energy of a particular wavelength except in UVB receptor, UVRB, that uses specific tryptophan residues from the protein for UVB absorption. Some photomorphogenic responses in plants are regulated by specific photoreceptors, while others are regulated by several light signals and different types of receptors working together or antagonistically. Now let us see the blue light response in plants. There are three types of flavin-based blue light receptors in plants that respond to wavelengths between 350 to 500 nanometer. They are cryptochromes, phototropins, and the jet loop family and UVR8. The cryptochrome photoreceptors perceive blue light as well as long wavelength UV radiation or UVA. They contain a flavin protein and a chromophore and are believed to have evolved from microbial DNA photolyase, which is an enzyme catalyzing light-dependent repair of UV-damaged DNA. The cryptochrome-mediated responses include the entrainment of the circadian clock, flowering, and photomorphogenesis. Next, the phototropins and the jet loop family of photoreceptors are another group of blue light photoreceptors that contain light oxygen voltage or LOV domain proteins. The phototropins also contain a serine or threonine kinase domain, which upon activation by light leads to increased kinase activity, and it results in photomovement responses such as phototropism, chloroplast movement, and stomatal opening. On the other hand, members of the jet loop family contain an additional F-box domain and several Kelch repeats, and they regulate the entrainment of the circadian clock and the onset of flowering. The third group of blue light photoreceptors are the UVR8 photoreceptors. UVR8 photoreceptor is a homodimer that perceives UVB light, and upon activation by the light, the homodimer breaks down into monomers that interact with downstream targets. Next, let us see about the red and far red light response in plants. Plants respond to red and far red wavelengths of light ranging from 600 to 750 nanometer in wavelength by photoreceptors known as phytochromes. Phytochromes are proteins attached with a light-absorbing chromophore consisting of a linear tetrapyrrole pigment called phytochromovilin. Phytochrome proteins is a dimer of two identical 124 kilodalton polypeptides having three domains with amino acid sequences similar to domains of other sensor proteins, and each polypeptide is covalently attached with a pigment molecule. The apoprotein of phytochrome is commonly synthesized in the PR form. The apoprotein becomes sensitive to light when it binds with the chromophore and changes its conformation to the biologically active PFR form when it absorbs red light. The PFR form can switch back to the PR form by absorbing far red light. There are multiple phytochromes in plants encoded by different genes. For example, 5 phytochromes are present in Arabidopsis, and they are phytochrome A, phytochrome B, phytochrome C, phytochrome D and phytochrome E. Phytochromes mediate important responses in plants including photoperiodic induction of flowering, chloroplast developing not including chlorophyll synthesis, leaf senescence and leaf abscission. Let us now see the characteristics of phytochrome-mediated responses in plants. First, the action spectrum of the light needed for phytochrome-mediated responses shows a peak in the red region at about 660 nanometer in wavelength. Next, light absorption by phytochrome displays a decrease in absorbance at 660 nanometer and an increase in absorbance at 730 nanometer, which is caused by the conversion of red absorbing form or PR to the far red absorbing form or PFR. The PR form shows absorption peak at 666 nanometer and is synthesized in dark grown seedlings. The PR form is converted to the PFR form when it absorbs red light. The PFR form shows absorption peak at 730 nanometer and is the active form that initiates biological responses. It can revert to the PR form spontaneously in the dark, also known as dark reversion, or when it absorbs far red light or in the presence of proteinases. The phytochrome-induced responses can be reversed by applying far red light of the wavelength 730 nanometer just after the red light treatment. Next, the levels of phytochrome are about 50 times higher in dark grown seedlings, mostly at the apex of the plant, than in light grown plants. The next feature is phytochrome allows plants to sense higher proportion of far red light when they are shaded, and the plants respond to greater proportion of PR to PFR by elongating stem and developing smaller leaves and less branching. The next characteristic is in seed germination. Seed germination in many plants such as lettuce seeds with low amounts of storage reserves require red light, while germination is inhibited by far red light. Such seeds require PFR for germination, and they do not germinate if they are buried below the level of light penetration in the soil or sown in places shaded by a leaf canopy. Let us now see the conclusion. To conclude, growth and development in plants are controlled both by internal as well as external factors. Plants respond variedly to these factors, and the responses govern the different physiological processes taking place inside the plant body. Plant hormones are the most important among the internal factors, while light is one of the most important external or environmental factors that have a major impact on plant growth and development. Light signal is perceived by specialized photoreceptor molecules in plants, and phytochrome is one of the most important photoreceptor that perceives red light. Light controls many aspects of plant growth and development, including photoperiodism and photomorphogenesis that control important processes like germination, growth rate and flowering, etc. Thank you.